Okay, so that was up to the part that I want to share a little bit about the background of these viruses and hopefully to level set why I'm interested in these and viruses in general and the need, I think, for continued ongoing research to really understand the basic mechanisms. So now what I want to do is I want to turn to looking at structural studies of the genome of the coronavirus OC43. So before I do that, let me briefly just sort of explain the difference between two forms of translation, i.e. making proteins that both of these viruses essentially exploit. So coronaviruses use what's referred to as a cap-dependent mechanism to regulate translation. And what that means is that on the 5' end of the viral RNA, there's a 7-methylguanosine cap. And that structure can be recognized by this initiation factor, EIF4E. This is a cap binding protein. And when that protein binds, it can assemble other factors to form what's referred to as a ternary complex that can scan along the 5' UTR to find the first AUG and then start protein synthesis. Alternatively, enteroviruses lack a 5' 7-methylguanosine cap, and they use what's referred to as an iris, which is a highly folded RNA structure within the 5' UTR of their genomes to recruit RNA binding proteins collectively known as ITAFs, other initiation factors, 4G, 4A, to then help assemble this 40S ternary complex, which can scan the 5' UTR, locate the first AUG, and then start protein synthesis. So these are two different mechanisms that are being exploited by these different viruses, cap-dependent and then cap-independent. So something that you should know about viral 5' UTRs in general, they are some of the most conserved regions within the genome of the viruses. So in addition to the 5' UTRs and the 3' UTRs, you see a lot of conservation in terms of structure and in some cases sequences. They often fold into very elaborate tertiary and secondary structures. And these structures are believed to do things like facilitate binding of the ribosome, facilitate association of other RNA binding proteins, and also aid in things like viral replication and packaging. So they're very functional units. So there's a lot of selective pressure on these UTRs to maintain their shapes and in some cases their sequence. As I mentioned, they regulate these key viral events, primarily translation and viral RNA synthesis, and they often recruit a large number of cellular RNA binding proteins. And the mechanisms by which they recruit these proteins and what they actually do, I mean, some of these things can be open for debate, but they are thought to fine-tune translation and viral RNA synthesis, via some balanced type of process. But the other thing that I think is important for us to recall is that these viruses, these positive RNA viruses, primarily replicate in the cytoplasm. A lot of these cellular RNA binding proteins are nuclear. So when they infect the cell, there's a shuttling of these nuclear RNA binding proteins, some of them involved in splicing, polyadenylation, other RNA processing events. Now they're inside the cytoplasm. So the viral infections are really reprogramming the cellular environment in many ways as a part of facilitating their replication cycle. So we started studying during the pandemic SARS-CoV-2, but we also started a project on looking at the 5' UTR of OC43. And what we like to do with any RNA we study is we have multiple tools in the toolbox. And because we're looking at viral RNAs, as I mentioned, there's a lot of selective pressure on their UTRs to maintain structure and sequence. So we can use phylogenetics to really kind of help us see if there's some evolutionary signal that's important to then seed a more long-term project. You've heard from Andrew about chemical probing. We use chemical probing as well to help us get some baseline structure on what the RNA might look like, and we've used DMS and shape. I'll show you some of that data. My lab is primarily an NMR lab, so we like to use NMR spectroscopy. And really what NMR spectroscopy does is it can kind of fill in some of the gaps that we miss by looking at just chemical probing only. We can directly visualize hydrogen bonds. We can get information about parts of RNA that are not base paired, but still have some type of structure that can be important, and also learn about dynamics really from a picosecond, the nanosecond timescale. I think I lost my PowerPoint. 1 second, let's see if I can really open that. That is weird. Hopefully this opens. Okay, great. Okay, let me just jump ahead to where I was. Okay. So we use NMR. We also like to get information about the global tertiary structure of the protein, of the RNA. And we're okay that, even if we get this information at low resolution, because what we try and do is integrate across different methods to create more, complete pictures. And so we use small angle X-ray scattering to get some type of low resolution global information on the structure of these molecules. Computers are essential to all the work we do for predicting structures, for running simulations, et cetera. And although I don't refer to people as tools, it's always nice to have good collaborators to help you make sense of the biology of the stuff that you're looking at. And just really to extend beyond the things that, you can do individually in a lab. So we collaborate pretty extensively. So what you're looking at here is now the 5' the structure of the 5' UTR from human coronavirus OC43. We probed this initially with DMS MAP-C, and you can see the reactivity index superimposed upon the structure. So this RNA is highly folded within about the first 330 nucleotides. It folds into 4 easily identifiable stem loops, and then a much larger multi-branch stem loop with several kind of stem loops emerging from this central junction. We also looked at this RNA by shape chemistry, which you heard Andrew talk about as well. And when we compare the shape-derived structure and the DMS-derived structure, the first 4 stem loops are pretty much the same. We have some additional base pairing that we can see here in this large internal loop, and we also validated that by NMR. DMS just didn't do as good of a job there at shape. But we also can see that this large stem loop 5 refolds into a slightly different structure than what we see when we just look at it at DMS alone. So this just validates, at least for us, that this region of the RNA can adopt multiple conformations. And we also saw evidence for that in the NMR as well, because the NMR signals just really got broadened out when we tried to look at it. We wanted to understand something about the conservation of this structure, but really the sequence epitopes within the structure. And so we performed a conservation analysis. And so the more conserved residues are in red, the least conserved residues are in purple. And for these first 4 stem loops, you can see conservation really, there's a high degree of conservation at the apical loop of stem loop 2 and at the apical loop of stem loop 3. And these stem loops are known to be involved in viral RNA synthesis. And so that's one of the reasons why they're likely highly conserved. What was also interesting to us is that the linkers that separate these different stem loops are relatively short. And so between 1 and 2, you have 1 nucleotide, between 2 and 3, 2 nucleotides, and between 3 and 4, 1. The length of those linkers vary slightly depending on if you're looking at the DMS structure or the shape structure, but nevertheless, they're really short. We combined NMR and small angle X-ray scattering to get a global 3-dimensional structure of this RNA. And what we observed in that structure is that stem loop 3 and stem loop 4, which are shown over here, coaxially stack to form this one long helix within the 5' UTR. And that allows stem loop 2 and really stem loop 1 to kind of like pivot, if you will, to some degree around this long coaxially stacked helix. What we thought was pretty interesting when we look at this tertiary structure, right? We've gotten rid of the multi-branch domain here, but the small angle X-ray scattering data suggests that this long helix is still there even when you have the stem loop 5. We see that these conserved residues now are more juxtaposed in three-dimensional space, and that's something you really can't appreciate just by looking at the two-dimensional diagram. We don't know if this juxtaposition of stem loop 3 and 2 are important, but they are both involved in viral RNA synthesis. And in the paper, we did measure a difference in binding affinity for the nucleocapsid protein, depending on when we had just an isolated stem loop 3 or the intact structure here. So there might be something there. So we wanted to go on and just ask, okay, is there a biological signal associated with that particular structure? So in collaboration with Shinru Shi's lab in Taiwan, we carried out these luciferase reporter assays. So the top reporter is our control. So we have translation that's driven by 5' cap. We really don't have any large RNA structure between the cap and the AUG that codes for the vanilla gene. For our constructs where we're looking at the dependence or the influence of the viral 5' UTR, we use the firefly luciferase, again, cap driven, but now we're going to drop in different constructs of the 5' UTR and see what happens. And so we're plotting out relative firefly to Ranella ratios along the Y as a function of different constructs that we use here. And so one of the very first things that you can note is that once you drop this large RNA in downstream of the 5' cap and you measure relative to the control, this large RNA structure is really reducing translation efficiency, right? So you're at about 0.2 in terms of your ratio. If you then delete this large stem loop 5 here, this multi-branch domain, and ask, well, what happens? You get a further reduction in the relative ratios of these two reporters. And so that would then imply, right, that multi-branch loop is acting as some type of enhancer to translation, right? Because you remove it, you get a decrease in activity. Now the question is, we really wanted to probe this coaxially stacked helix that we observed. And so what we did, we engineered mutations that will disrupt this lower helix that's forming here, right? So we disrupt those Watson-Crick base pairs. And we introduced a second set of mutations where we increase the length of the linker between stem loop 2 and 3. And so when we disrupt those base pairs, now we get an increase in the relative ratios of translation compared to the deletion construct alone. But what's really interesting is when we insert a long linker between stem loop 3 and stem loop 4, now we get levels of translation that are even higher than when we had the intact construct itself. So what we think this means is that this coaxially stacked long helix is acting as some type of barrier for the ribosome to move through. And if you increase the length of the linker residues, such that now you no longer have those stem loops coaxially stacked, now the ribosome can move through that RNA structure with a little more ease to make viral proteins. All right, so and the last part here, what I want to share with you about is our work to really try and inhibit enteroviruses by targeting structures within their 5' UTRs. And what I actually think this is kind of enlightened me on personally is that small molecules can be really useful chemical biology tools to probe protein RNA complexes. And so a place that we're moving into in my lab is really trying to do just that, to have a an assortment of small molecules that we can, look at a protein RNA complex and try and send it down a predictable pathway. So the enterovirus 5' UTR consists of 6 highly conserved stem loops. Stem loops 2 through 4 is known as the iris, or the internal ribosomal entry site, and it's of the group 4 sort of flavor, if you will. So these types of irises, they lack a lot of long-range tertiary structure, and so the dogma is that they bind a large fraction of RNA binding proteins to help compact the structure such that the ribosome can load and then start translation. I don't think that's the only thing that these RNA binding proteins do. I think there's other sort of biological outcomes there, but that's kind of the dogma. Stem loop 2 is the smallest of the stem loops within the 5' UTR, but it binds the majority of the RNA binding protein. So what I'm showing you here, this one stem loop associates, that we know of to date, with binding 7 RNA binding proteins once it infects the cell. In addition, this stem loop also binds a viral-derived small RNA. So it's kind of like a microRNA that gets processed when this virus infects a cell, and this stem loop also binds that viral RNA. So there's a lot of selective pressure on this one stem loop, given that it binds so many different cellular RNA binding proteins, and it has to bind a viral-derived small RNA. So we asked the question, well, how conserved is this particular stem loop? And we performed some alignments several years ago now. And what we identified is that the ability to fold into this type of secondary structure where you have a 5 nucleotide bulge and a 7 nucleotide apical loop is highly conserved. We do observe compensatory substitutions in the helical regions, meaning that, there's more pressure to main structure there, because you can substitute a GC for an AU Watson Crick pair as long as the A-form helical geometry is retained. But in addition, right, we see high selective pressure on these unpaired regions in this bulge and this unpaired region in the apical loop, implying that those sequences must be important for either binding some cellular RNA binding protein, or perhaps involved in long-range tertiary interactions in the RNA. So we know through our studies and others that HNRPA1 binds to this UAGG element, and you heard Andrew mention in his last talk about HNRPA1. It's a pretty ubiquitous RNA binding protein involved in splicing, but viruses hijack it often. And when HNRPA1 binds to this UAG element, it upregulates translation through mechanisms we don't understand, but we know that's the phenotype. AUF1 and HNRPA1 compete for the same binding site within this bulge loop. And when AUF1 binds, it downregulates translation, again, through mechanisms that we don't fully understand or appreciate. But we know these two proteins are acting in an antagonistic manner to maintain some type of homeostasis by which translation is occurring in the cells. So the question is this bulge loop important for viral replication, right? Given that we see this high conservation of sequence, we know that it binds these different factors. So we mutated that central UAG element, which is the core binding motif for HNRPA1, to a CCC. And when we do that, what we observe, right, in this dual luciferase reporter assay, so if we now, in this particular construct, we're looking at an iris. So we can put the firefly and the runella in the same construct. The runella is driven by a 7-methylguanosine cap. The firefly is driven by the iris that we put in between. And then if we measure relative ratios of firefly to luciferase versus what we're looking at, you can see that the iris is as good as the cap with translation, right? Because these ratios are about the same. But when we engineer in these 3 mutations here, now the efficiency of translation for the iris drops to about 20%. So those 3 nucleotides within that bulge are very important for driving translation. We can also look at viral replication. So we have now viral replication as assayed via plaque-forming units on the Y as a function of time on the X. The wild-type virus is growing with very rapid kinetics, and then it plateaus off at about a 10 to the 9th PFUs. If you take this mutant that has these 3 mutations in this bulge, that mutant grows with very different kinetics. There's a longer lag, and then it plateaus at around 10 to the 4th PFUs. So that's about a 10,000-fold difference in titers just by engineering those 3 mutations in that bulge. And that's the only place right within the iris that we did. So what can we learn if we compare these 3-dimensional structures of the mutant and the wild type? We did this by combining NMR and SACs, and I'm skipping over all of the structural work that went into this just to focus in on the business end of the molecule. So the UAG, right, is in this bulge. In the structure, the backbone almost takes a 180-degree turn, 5' to 3' around that bulge. These nucleotides are highly stacked. There's really some significant body of NOEs that NMR measurables that we've obtained to really verify the stacking geometry here. If we introduce those CCC mutations, now you see that the backbone is a little more elongated. These nucleotides are more dynamic, and this is a more open structure compared to here. So this compact structure suggests that we have a pocket, at least some part of the time, that can be targeted with small molecules. We know genetically that those mutations in the bulge are important from our biological assays. So that was the question: can we target this bulge loop with small molecules? And we collaborated with Amanda Hargrove, who's at Duke University and has a library of small molecules, which are known as dimethyl amilarides. They all have the same core scaffold in red with different functional groups coming off that scaffold to give them, different type of chemical properties. So in collaboration with Amanda, we identified 6 compounds that have affinity for this bulge loop. By NMR, we validated that all 6 bind, but they bind with different mechanisms. The NMR signals go different places and they change in different ways. So that's interesting in itself. Only one of those molecules, DMA-135, however, had biological activity in the luciferase reporter, as well as in our viral replication assay. Importantly, that this small molecule does not affect cap-dependent translation. Cap-dependent translation is perfectly fine, even as high as a millimolar of the molecule added. We measure a CC50 around 100 micromolar and an IC50 around 7 micromolar. The KD for this molecule and the RNA is around 2.5, 3 micromolar, so the IC50 and KD are on the same order of magnitude. We can also dose dependently decrease viral titers by increasing the concentration of that small molecule. So we want to know, how does a small molecule work? Like, what's the mechanism? We went back to NMR. We saw the NMR structure of the small molecule bound to the RNA. And when we observed the structure, there's a lot of fuzziness here because we don't have a lot of NMR data to lock things down and unique conformations. But what's important is that this bulge, which was highly compact when the small molecule wasn't there, now it's floppy, it's dynamic. So we liberate some residues in the process of this molecule binding. Also, interestingly, if you compare the bound form of the RNA, which is in the surface representation, to the free form of the RNA, which is in the cartoon representation, when the small molecule binds to this bulge, remember those nucleotides are tightly packed, but now the molecule has to jettison its way into that major groove and liberate those nucleotides. And so in the process of doing that, now you get this roughly an 80-degree bend of the upper helix relative to the lower helix because the molecule is disrupting those stacked residues. When you do this type of work, right, if you really want to know if your molecule, your small molecule is hitting the target that you believe it's hitting, then one way of validating that with viruses is by performing serial passaging to see if you can select for drug-resistant mutants that then map to your target. So this is a very powerful assay that virologists use for many different reasons, but as it works out for antivirals, this is pretty powerful to say, okay, I know that I'm hitting my target in cell. There might be some other targets, but at least that one got hit. And so we carried out serial passaging in SF268 cells in the presence of 50 micromolar DMA135. You're looking at viral titers here on the Y as a function to passage number on the X. And so when you have 50 micromolar of the drug, you can lower the titers to about less than 10 to the fourth. You don't completely inhibit the virus, right? You want just enough virus around that through multiple passages you can select for those drug-resistant mutations. And so after 9 passages, right, we can identify viruses that replicate at levels similar to the wild-type virus without the drug. So again, back at 10 to the 9 PFUs. If you harvest those viruses and do a sequence analysis to see where the mutations are, you can see that we map mutations to this A now is a C and the mutant, and this C becomes a G and the mutant. And so what happens here, right, you disrupt 2 Watson-Crick base pairs. So this AU, which actually is highly conserved, by the way, I forgot to mention that this AU pair and this AU pair are highly conserved. It's never a UA, it's always AU, right? So this sequence here, this AAU sequence is important, okay? But nevertheless, you disrupt 2 base pairs in this resistant mutant. If you ask the question, how does this resistant mutant work to still drive translation? You can compare the wild type reporter to the mutant reporter, and it's almost just as good. So the mutation doesn't affect translation, iris-dependent translation. If you then ask, can the drug still work to inhibit translation? The answer is no. So here's the drug with the wild type RNA, and you see iris-dependent translation is way down, but here's the drug with the reverting only these nucleotides here, right? And the levels are comparable. So those 2 nucleotides are sufficient to confer resistance, drug resistance to DMA135, at least in that assay. We also then engineered viruses that only contain those mutations. And I want to emphasize only because this is important, because when we sequence the resistant viruses, we didn't do a full genome sequence, right? Because I can't remember exactly why we didn't do it. I think there was something else associated with cost, so we just really wanted to know the answer at the time. But we only sequence the 5' UTR, right? So there's the possibility that you can get some secondary mutations far downstream in the genome that are a result of this drug resistance. So to work around that, right, we just then engineered viruses that only contain these mutations, and then went back and did the assay. And you can see for yourself, by comparing to wild type drug with the wild type, levels drop significantly. Those engineered viruses are as good as the wild type in the presence of the drug. So we know that those 2 mutations are sufficient even within an infectious clone for the drug resistance phenotype that we observe. Now, how does this work? I mentioned that stem loop 2 is this regulatory axis. It binds all these different factors. So might these molecules actually be perturbing this sort of like equilibrium between these RNA binding proteins? And the answer is yes. I'm not going to show you all that data. It's in papers that we publish. But what the small molecule does is it allosterically increases the binding affinity for AUF1, which naturally represses translation, while leaving HNR and PA1's binding affinity unperturbed. But what about the drug-resistant mutations? Do these drug-resistant mutations affect how these factors bind? And so for HNR and PA1, the answer is no, not significantly. This is calorimetry. We're looking at heat exchange and the binding reaction as a function of molar ratio of the protein to the RNA. HNR and PA1, which activates translation, binds to this RNA with this characteristic biphasic transition. And the reason why there's an AUG element here. And then there's a second YAG element here, and we validated before that the protein can bind both sites. We think this one is really the one that's functional for translation. I'm not sure what the second site does, but that's the profile that you get. AUF1, on the other hand, cannot bind this reverting mutant at all. It binds the RNA as a one-to-one complex with a binding affinity constant around 300 nanomolar. But now when we mutate out these two residues here, one was a C, one was a A, we lose binding affinity. And we know from additional studies that we did in this paper that this A right here is a primary determinant for AUF1 binding. And so when you mutate that A to a C, now you really do lose binding affinity, as detected by calorimetry. We can also validate this in a more cellular context. If we transfect biotinylated wild type SL2 and the mutant form of SL2, dose in the drug DMA-135, and then go fishing for AUF1 and HNRMPA1, what we see is that we dose dependently increase the binding affinity for AUF1 for the wild type RNA. No binding is detected for the mutant. We can also see that binding of HNRMPA1 is retained both for the wild type and the mutant. Okay, so this really demonstrates that this molecule has selectivity at perturbing the AUF1 SL2 complex, but not affecting the HNRPA1 SL2 complex. So just kind of in wrapping this all up now, again, I hope that I convinced you that, or I hope that I didn't need to convince you that RNA viruses are, really a major concern for global and economic health. And they do this in part by how they replicate within the cellular environment. So just understanding basic biology is still important to understand sort of the battle that exists between viruses and their hosts. I hope I convince you that structures within the 5' UTR coronaviruses have properties that you might not appreciate a priori, but nevertheless, those properties are there. And in this case, forming this coaxially stacked helix to downregulate translation, and that we can actually inhibit viral replication via small molecules. And so that's really summarizing those two results here through looking at these two different structures that I shared the background work with you in the talk. Again, within the toolbox are people, and they've had a wonderful trainees over the years to work on these projects. They've all gone on to successful careers in biotech, and one is working on his MD PhD at Penn State. And then just amazing collaborators for helping us with the chemical biology and helping us with the virology. And then I would also like to thank my research group, and then these other 3 members here who did the coronavirus work that I shared with you, who were just not, I didn't have headshots to put them on the last slide.